mentioned earlier is going to be about hematopoietic stem cell transplantation with a focus now on allogeneic stem cell transplantation. And we have three outstanding speakers. The first speaker will be Dr. Richard Ambinder from Johns Hopkins School of Medicine, and he will talk about allogeneic bone marrow transplantation with optimized antiretroviral therapy.
Great. Thanks. I want to thank the organizers for inviting me to this wonderful meeting. And if you've spent your life in bone marrow transplant, it's kind of fun to get invited to Seattle to talk about bone marrow transplant. And on the other hand, I see a very mixed audience with very different backgrounds in bone marrow transplant. So if you're a native of the Fred Hutch, forgive me if I am mentioning things that are already very familiar. But let me begin by saying we're going to talk about the possibility of moving towards cure in people who are getting bone marrow transplants. But as you've heard, transplant remains a nasty business, although not nearly as nasty as it used to be. And in fact, a world different than it used to be. But I don't think there's anyone who's involved with transplant who's advocating transplant as an approach, advocating allotransplant as an approach to people who don't have a malignancy or another fatal disease that requires the intervention. My partner in crime here is Christine Durand, who's a faculty member at Hopkins in infectious disease and works with the Silicano group and is a fellow of the AIDS Malignancy Consortium. But I'm going to talk about data generated from a much larger Hopkins group, and they're just not all recognized as authors here. The story for us at Hopkins with transplant and HIV actually begins long, long ago and far, far away. If you can remember back, those of you who are old enough, to the time when the Soviet Union was withdrawing from Afghanistan because it was such a complicated place. That year, the Hopkins group had a patient with HIV and Burkitt's lymphoma. 1988 was the transplant, and AZT had just been recognized as having utility in the treatment of HIV and was the only antiretroviral available. And with the evidence that HIV had its reservoir in hematopoietic cells, the patient was transplanted under cover of the antiretroviral agent. And just to remind you how long ago it was, I need to show you HIV PCR from 88 and 89. The PCR was different than what we have today. It didn't show any residual HIV in the patient. The patient died of relapsed Burkitt's lymphoma. The autopsy by first generation PCR was clean of HIV. There were a series of transplants done that had similarly bad outcomes for a whole variety of reasons, not least of which was transplant was still pretty ugly and we didn't know much about supportive care for HIV patients and so on and so forth. But later, Christine and I talked about, gee, couldn't we bring this approach to our transplant patients? Couldn't we protect donor cells from getting infected? And as you've heard or you'll hear, the group in Boston anticipated us, but we didn't know that at this point. We were thinking that no one else was doing this, and put together a protocol to take patients who needed allotransplant for malignancy and to ensure that they didn't miss any antiretroviral therapy, with the idea that a lot of patients have nausea and vomiting during the course of transplant, or have bad mucositis or something else that interferes with their ability to take antiretroviral therapy, and that if you were missing doses, that might be enough to allow donor cells to get infected, and then no matter how adherent you were after that, you'd already lost the battle. And we spent a lot of time working on minimizing bad drug-drug interactions with the multitude of drugs that are used in transplant, and trying to optimize regimens as people came to transplant. And finally, and we're going to talk a little bit more later, using enfuvirtide, which is the only, as of now, approved, aside from AZT actually, approved antiretroviral therapy that can be given parenterally. And on the days that people were getting high-dose cyclophosphamide, which is a key part of our regimen 3 and 4 days post-transplant, that they would, even before they started vomiting, on the chance that they might be vomiting and might be missing doses, that they would have enfuvirtide added to their regimen. So we had fairly modest goals in the study. Would enfuvirtide be tolerated during transplant? Could we actually keep antiretroviral therapy going? We have fancy electronic drug bottles so that we can tell whether a patient's open their pill bottle every day, and not that you couldn't get around it if you wanted to trick the bottle, for sure you could, but gee, there are an awful lot of people who don't open the bottles and aren't trying to trick anyone, but if you just ask them, we'll say, oh, I haven't missed any doses. So we've now transplanted 6 patients who are listed here. There's one patient, number 5, who died before he ever got transplanted. 4 of the patients are alive. 2 of those are mixed chimeras. 2 of those are complete chimeras. And the last 2, patients 6 and 7, I should say, are haploidentical related transplants. So again, for the non-transplanters, the standard until a few years ago was that people were getting only fully matched or very close to fully matched transplants, and the national CTN-AMC protocol requires a full match or a single antigen mismatch. But we've for a long time been doing haplo-matched transplants, and now there are at least 8 centers that have published that haplo-related transplants are as good as fully matched related transplants, and our haplo patients are doing well. Enfuvirtide, also called T20, was well tolerated. There were no issues. Not all the adherence data is shown here, but some people were very adherent. Some people were not at all adherent. And so enfuvirtide can certainly be used. In terms of the viral outgrowth assay, and this was done in the Silicano lab, all of the patients who got complete chimerism had evidence of their reservoir by the viral outgrowth assay disappear. The 2 of 6 who had mixed chimerism, that is 20% patient cells persisting, still had a reservoir that was readily measured by the viral outgrowth assay. So while we were doing this, the whole Boston story evolved, and I think the messages are very similar. What's different is that we have a lot better data in terms of when and whether people actually got all their doses. We have a lot more early time points. And I'm going to tell you a story. So the Boston group reported viral rebound in the 2 patients who were deliberately taken off antiretroviral therapy. We haven't deliberately taken anyone off antiretroviral therapy, but we have a patient who took himself off. Let me tell you his story very briefly. An acute leukemia patient who actually had some history of noncompliance that led to multiple relapses, making him ineligible for the national allo study because he was had so many relapses. Went through transplant with post-transplant Cy on our protocol, got his marrow from a matched allo donor, achieved 100% donor chimerism, had undetectable HIV, and then stopped taking his medicines, it turns out, got fevers and issues that we didn't understand for a while, eventually got admitted to the hospital, sank into stupor. His first days in the hospital, he was refusing to take his antiretrovirals. He got a lumbar puncture and had a CSF viral load that was almost as high as his plasma viral load. He was started on enfuvirtide, woke up, signed out of the hospital AMA. And like the Boston group, we learned that rebound with a meningoencephalitis is a pretty frightening thing, and that there may be some special risks for people who have HIV, who undergo allotransplant for treatment of their leukemia or lymphoma because they're getting donor cells from an HIV negative donor, and if they're getting full donor cell engraftment, there may not be any T-cell response to control the HIV when therapy is interrupted for one reason or another. I'm going to very quickly show you some phylogenetic data that attempted to say where did this relapse come from. Was the CSF reservoir different than the plasma and different than what was there before transplant? And there are several time points that were looked at, and bulk sequencing was done with next-gen sequencing. And I won't belabor the details except to say that it appears that the CSF relapse was from the same population of cells as had been in the peripheral blood initially. There wasn't any evidence for a separate reservoir. So again, doing transplant in HIV patients may have some separate risks. And again, for people who do marrow transplant in general, this is kind of a familiar story. This is what happens with CMV. If you have a patient with CMV and they get a CMV negative donor, they're at risk for developing CMV post-transplant, in part because there are no donor cells that know how to deal with CMV. And as initially pioneered in Seattle. There are approaches to adoptive cellular immunotherapy that seem to control CMV if you can give CMV-specific T cells in the post-transplant setting. So future directions for us, we're very interested in the possibility that you may be able to protect HIV patients going through transplant from what could be a fairly dangerous rebound by using adoptive immunotherapy with donor-derived HIV-specific T cells. And Kath Ballard at Children's Hospital in DC has been making HIV-specific autologous T cells for a clinical trial in North Carolina from again, seropositive patients, but has recently reported that she can make HIV-specific T cells from HIV-naive donors pretty consistently, and we're working towards testing that out in a phase 1 trial in patients who are going through transplant for other reasons to see if these cells are well tolerated, if they have side effects, if they cause GvH, and if they may ultimately interfere with rebound and perhaps help clear the reservoir. Let me spend just a minute then saying, why has the Berlin patient not been repeated? And I think you've heard the story that gee, it's hard to find a perfectly matched donor who's also CCR5 delta 32. It's like waiting for lightning to strike twice. We actually had the experience of being ever so close in the national CTN-AMC trial. We had a patient who had a dreadful lymphoma who finally went into remission, and we enrolled him in the trial and sent his HLA data off to the NMDP, and we got a call back, this guy's got thousands of potential donors. We said, well, great, let's pick the one that's CCR5 delta 32 homozygous. And they said, well, we haven't done that typing yet. It's going to take us 2 to 3 months. We have to call all the donors, we have to get permission, we have to get blood or cheek swabs or whatever, and we told him he couldn't wait, and he went ahead. He's continuing in remission now 2 years, but almost certainly would have had such a donor if he could wait for all the typing to have been done. We've known for a long time cord blood doesn't require this kind of stringent matching, and cord bloods are banked and ready to go, and this is an old slide that probably should be updated, but a lot of cord blood units have been identified that are homozygous. A few patients have been transplanted. The outcomes that I'm aware of are not yet good outcomes, but it's early, and I'm sure there are going to be some successes using cord blood. In Baltimore, we've drunken the Kool-Aid that we call post-transplant Psi, and we don't think matching is very important in general anymore. And I've told you that our data, and now many other places have reported that haplo and fully matched donors do the same if you use post-transplant Psi. Yvette Kassaman at Hopkins said, well, what about unrelated donors who are mismatched? Can we use substantially mismatched donors and get away with transplant? So this was not an HIV question, this was a transplant question, but we've done 16 patients who were unrelated, serious mismatches, with 3 of them being 7 out of 10, one of them being a 6 out of 10 match, and one of them being a 5 out of 10 match. And for those of you who obsess about HLA, 10 of the 16 were mismatched at HLA-C locus, and there was a DP mismatch in 13. So these are seriously mismatched patients. All the patients engrafted. There were no non-relapse deaths. There were no prohibitive toxicities. There were no diagnoses of acute grade 3 or 4 GvH. And so we think that unrelated mismatched donors could be used. And are there unrelated mismatched donors? Of course there are. There are 20 million people in the registry. Most of them, the great majority, haven't been typed, but in fact the Germans, after the Berlin patient, have gone ahead and started typing people as they join the registry now, and they've typed more than 1 million donors, and so have identified probably about 10,000 donors who are HIV resistant. And if you're willing to accept some mismatch, I think 30, 40, maybe 50% of Caucasians, and a lower but still serious fraction of non-Caucasians could have mismatched unrelated donors who are HIV resistant identified. And we're interested in pursuing that as well. Again, many people contributed to the data that I'm showing you. I've mentioned a few here, and I'm good for questions. Yeah.
Thank you very much.
Can you tell us exactly what you mean when you say 100% donor chimerism? Presumably it's blood. What do you mean that looks at other compartments? And then also, what's kind of the maximum amount of donor cells could be persisting that you're simply not able to see in the assay?
So it's a great question, and right now we're only using the standard clinical assay. So what we mean is that there are less than 5% patient cells persisting, or in some cases less than 1%, but we haven't done the very fancy assays yet that will get down to lower percentages. We've got specimens saved, so we should be able to do that, but we haven't yet. And marrow or blood, we have counted either. They're sometimes discrepant, but in the people who are seemingly complete chimeras, we're seeing the same in both.
I'm intrigued by your comment that HIV may reactivate in the setting of immunosuppression, and that makes me think back to Zach's first lecture about whether the latent reservoir is not just purely determined by the drug therapy but also residual anti-HIV immunity in patients. That is, the latent reservoir set point is determined not just by drugs but also by whatever that patient's immune system is doing to control the virus. And do you think?
It makes a lot of sense to me, but I don't have any special insights here. But along those lines, let me mention that it was suggested that people who got transplant have to remain on immunosuppression for life, and our patients come off traditionally at 6 months, but in our more recent protocol at 60 days. And so it's not a lifelong immunosuppression, it's immunosuppression for several months to a half year if they're not having problems with GvH.
The other problem you really hit was that this is completely naive to HIV. So the immune system is completely
Right.
naive to HIV. So the question was whether there's any advantage to some sort of therapeutic vaccination or some of the maybe gene therapy approach Dr. Baltimore's going to talk about, or something of that nature that would be done in parallel with this that would be useful.
Yeah, so I think I think for sure that's a direction that we're going to be moving. I think the whole thing about vaccinating donors is quite a can of worms and not likely the place to start, but the idea that you might add an antibody or add T cells that are targeting HIV is very attractive.
You mentioned cord blood as an alternative to matching. Can you talk about maybe the feasibility of doing editing on cord blood and how that would fit into a transplant scenario?
So let me back up and say that that cord blood has been a great success in children, and in adults, much of the transplant world is divided into those who use post-transplant Cy and mismatched donors, and those who use cord blood. I'm from the home of the cord blood doubters. Very briefly, the concerns about cord blood in adults are that cord blood, sort of by definition, does not have mature T cells. And in order to make mature T cells, you need a thymus. And if you're a real grown-up, you don't have a thymus, and you don't have thymic function, and that one of the problems with cord blood is never really recovering immune function. Now, there's a randomized trial going on internationally comparing cord blood and mini-haplo with post-transplant Cy. So, my prejudices soon should be replaced by real insights into the differences, and we should learn whether that's true or not. But we don't see problems with chronic infections with post-transplant Cy at a first approximation in most patients.
I would probably comment that still I think both options are out there, cord blood versus haplo. I mean, cord blood certainly there's probably the immune reconstitution requires a bit longer, but at the same time there's also donors available for everyone, and typically less graft-versus-host disease. Haplo, I do think you probably still see a bit more graft-versus-host disease than compared to haplo.
We see less graft-versus-host disease than cord blood, and the CTN trials that were published together comparing mini-haplo and cord blood showed less graft-versus-host disease in the mini-haplo arm. But we'll see in the randomized trial.
Exactly. So I think both options are still out there, especially for adults.
For sure, both options are out there, and we're participating in the randomized trial.
So, go.
Just quickly, you should be aware there's a pediatric impact ACTG study that allows cord blood transplants with homozygous CGT4HIVCure. It will also allow a haplo transplant, and there apparently is one such patient in New York that has been treated. Can't tell you the stuff.
So I did have one additional question.
Yes.
So, obviously, your center, we have done about 10 transplants now with non-myeloablative conditioning in these patients. So we can do the transplants, allogeneic transplants, whether unrelated or haplo. But the question is going to be, when should we offer, can these patients come off potentially ART therapy? Is there any consensus or an effort to maybe work together and figure out what is a good parameter to then suggest having patients undergo a structured treatment interruption?
I think you're just making the transition to the next speaker, and I'm going to let him address this question.
Okay, wonderful. So, our next speaker is Leslie Kean. She is now from Seattle as well, moved here just a few years ago. She is with Children's Hospital and the Fred Hutch, and her title is Allogeneic Hematopoietic Stem Cell Transplantation, HIV Eradication. What can we learn from translational models of graft-versus-host disease?
Great. Great. Super. Thanks, everybody. It's a real pleasure to be here and talk about our data from my lab. So we became interested, so I'm a pediatric bone marrow transplanter clinically, and I've been interested in the graft versus host response for many years. And several years ago now, with the discovery of the Berlin patient, we became interested in how to tie these two fields together, HIV cure work and graft versus host disease. So I think there are still many more questions than there are answers, but my lab is now really focusing on what graft versus host disease can potentially teach us about the graft versus reservoir effect. So we've gone over this a lot this morning, but what we know about the only patient that's cured of HIV is he, like we've said, he's had a complicated treatment course. So he received not one, but 2 myeloablative unrelated donor stem cell transplants. He developed graft versus host disease, we know that, and he was transplanted with stem cells from a donor that was homozygous for the CCR5 delta 32 deletion. So the relative effect of each of these parts of his treatment are still really unknown, and one of the things that my lab is doing is really focusing on the graft versus host disease and seeing what we can learn about how that process might impact cure. So as I said, there's more questions than answers in dissecting the necessary and sufficient components of a transplant-based cure of HIV. And the specific questions that we're focusing on are, number one, what are the relative contributions of the many elements that the Berlin patient had? Is there a graft versus HIV reservoir effect? We assume that there is, but really this has not been shown yet. And how do we measure it if we can? And of course, the ultimate goal for our patients is, can we recreate that sterilizing cure that occurred in the Berlin patient? This may not be relevant to every single all-comers with HIV, but if we can show in those patients that need transplant that we can cure them, that's a major proof of concept for the field as a whole. So I've taken an approach of studying this problem in a translational model of allogeneic stem cell transplant that I created in Rhesus macaques many years ago. This model really requires both clinical expertise in bone marrow transplant and also a really robust system to study this in a very translational system. We use the Rhesus macaques. We are now very closely collaborating with Hans Peter's group here, starting to do experiments in pigtail macaques as well. I was at Emory until a couple of years ago. I worked both at the Emory, the Yerkes Primate Center at Emory, and now I'm at the Washington National Primate Center. To do these experiments, you need to be able to recapitulate the clinical milieu of transplant for our patients. So you need a fully operational OR, you need dedicated leukapheresis because you need to get those stem cells for your transplant recipients. You need capacity for TBI. Here at the Hutch we have our own research irradiator, which is really very important for getting our animals through the conditioning. You need indwelling catheters, just as we do for the patient. So it's very complex. Most importantly, you need expert pathologic analysis, because I think if we're going to use this model to really understand mechanism, what we really have to capitalize on is our ability to really look at that reservoir in more than just the blood and the bone marrow, where we're often limited in patients. And so that's really what we're driving towards in the work that we're doing here with Hans Peter. So we've talked a lot about haplotransplant, we've talked about cords, we've talked about NK matching and HLA matching, and one of the things that my lab did several years ago now is to bring the non-human primate field up to where we were clinically and up where the mouse field was and the dog field was for many years in terms of getting MHC-defined transplants. So if we're going to really study the graft versus reservoir effect, we need to be able to dial in MHC disparity at whatever level we want. So we've now done this. We've typed colonies. There are now several colonies around the country that are typed. The Yerkes colony is now typed. The Washington colony is now typed. We're working with Davis to type their colony, and NIAID actually owns a colony that's now typed. So more and more, these are really feasible experiments. And what you can see here is just an overlay, a color code that I created on a colony we typed many years ago, and we published our sort of methods in 2011, where the color code of the different MHC chromosomes is shown for each animal. And what you can see is now we can dial in our MHC disparity. So if we want to do a matched transplant on our animals that are infected with HIV, we can. We can choose matched siblings, here as an example here. Very importantly, we can do haploidentical transplants. And in fact, that's the model that I'm going to show you today. Not only I think it's really relevant for our patients where there are a lot of haploidentical pairs out there, but in terms of what we have available in our colonies, because of the harem-style mating that macaques do, where one male mates with several different females, you have tons of haploidentical half-siblings in any colony. So this becomes a highly feasible experiment. From any 20 animals that I type, at least 10 are going to be potential matches for my program. And then you can also do unrelated and any degree of HLA or MHC disparity there. So that was a big hurdle that the field had to overcome to really start modeling this in Rhesus macaques, and so that's really been done now. And so now we've developed a full model of graft versus host disease in the primate, and we're starting to overlay this with the HIV reservoir studies. So when I say full model, we really need to be able to dial in every single one of the variables that we see when we're doing patient work. So I just talked to you about the ability to dial in the MHC disparity, and we've overcome that. As I said, I think it's really necessary when you're doing these experiments to be linked to a National Primate Research Center. Really, I think those are the centers that are best equipped to do the conditioning that we need for our animals. So we can do myeloablative conditioning. We can do what we call in transplantation RIC conditioning, or reduced intensity conditioning. And we can really study the impact of the conditioning on the reservoir. And I'll show you some data that we published last year in PLOS Pathogens about that. And then I think, what we really focus on is the impact of graft versus host disease, or the T-cell activation that happens after transplant on the graft versus reservoir. And we can also dial in any of those. We can use our standard post-graft immunosuppression therapies, tacrolimus, methotrexate, post-transplant cyclophosphamide. So we can essentially model any of the graft versus host disease regimens that we use clinically. And finally, and really most importantly, we can do this detailed immunologic analysis of immune reconstitution, function, viral activation, and viral reservoir, and that's really what we're driving toward here. So several years ago, in very close collaboration with Guido Silvestri when I was at Emory, we decided to first focus, and I think one of the strengths of doing this in the primate model is you can really break down each of the steps here. So we decided first to focus on the impact of conditioning on the reservoir. And what we found was very interesting. So we set up actually a very compressed infection and ART treatment strategy. This was the first time that this had been done, and it was actually very unclear at the time whether we'd be able to get animals through transplant, just as we've been worrying about with patients on ART, et cetera. So we infected animals with the RT SHIV. We then treated them, they established infection. We then treated them with ART. We had, previous to the infection, so we were transplanting them in a situation that would, which would model a transplant with an uninfected marrow or stem cell source. We had, previous to the infection, we had collected their stem cells so that when after they were on ART, we could do a transplant and test the impact of, in this case, total body irradiation on the reservoir, giving them a marrow that had never seen HIV. So this actually gets to, and we've been discussing today, what are the relative benefits, and really most importantly, what are the relative risks of giving a marrow product that has never seen infection. We then kept them on ART through reconstitution, and then we did a planned treatment interruption. And as I said, we published this now in 2014 in PLOS Pathogens, so I won't go into lots of details. You can read it in the paper. But what we found was very interesting. So here are our transplanted animals, and then we did a control cohort here. So you can see that they had high viral loads that were controlled with ART. These are 3 animals. They all controlled with ART. Here is our transplant in green. Here is our non-transplant animals below. What you can see is very similar to what we've seen recapitulated in the patients, is that right after conditioning, you see these blips in all of our animals. And I think that's really important. It's one of those things that you're seeing in patients. I think this is probably, first of all, a window of risk for patients with HIV, but also a window where you probably could do vaccination or therapeutic cellular therapy. So I think this is a really important observation. These animals stayed, and I'm just showing you the RNA here, but actually their DNA levels actually dropped below what we could measure in the non-transplanted patients, but they stayed under relatively good control until we released ART. In the non-transplanted patients, as we would expect, as soon as we released ART, they had viral rebound. In the transplanted patients, they had not just the same viral rebound, but to higher levels. And these are results that Hans Peter has now shown in his pigtail macaques too. And again, I think this goes towards that question of risk. If you're going to do a planned treatment interruption in someone with a transplant, before they have fully reconstituted their CD8 immunity. So here we took them off ART about 100 days after transplant, and their CD8, even their quantitative CD8 immunity is not back to where it was. Certainly their qualitative CD8 immunity, and we've done this in other studies in our lab, is not up to normal. And what we saw, and these were short-term experiments, we saw that compared to the non-transplanted animals, the viral rebound that we saw in our transplanted macaques looked very similar to what we've seen in patients, in that it rebounded to really a much higher level. These animals got very sick, and there is an implication that there is an active CD8 immunity that you lose after transplant. You've got to think about this carefully when you're doing planned treatment interruption. So I think our results were very important in terms of beginning to understand the impact of transplant conditioning on the reservoir and what it will and will not do for viral load after our interruption. So we've now gone back and are doing a large series of experiments, putting the same experimental regimen through with an allo transplant. So we've got many animals now infected. They're starting ART, and these are long-term experiments, of course, and we'll have hopefully the first data on those in the next year. But as we wait for that data, what we're trying to do is understand as much as we can about the T cells that are going to impact the reservoir potentially as part of that graft versus host disease response in what we think is a graft versus reservoir response. So I'm going to show you some interesting data that we've gotten relevant to the HIV field from our non-infected transplant experiments. So we've done MHC haploidentical transplants, very similar to what's being done around the country. We're using myeloablative conditioning, and here we're using standard graft versus host disease prophylaxis. So what we see when we do this, we've now done many, many animals with this, and we have, I'm going to show you results here from 3 cohorts, and some more in the future in later slides. But so we have the ability in the non-human primates to do basically the natural history of graft versus host disease. So something you obviously could never do in a patient. We can give recipients a transplant without any prophylaxis, so we can really see the natural history of the graft versus host response. We can then compare that to both standard and novel therapies. Here I'm going to show you the standard therapies. We can do rapamycin monotherapy, tacmethotrexate as well. And we can not only monitor their donor chimerism, so all these animals become full donor chimers to the extent that we are measuring standardly, and I think it's important to really measure this in very sensitive assays as we do in the HIV-infected animals. We can look at their GvHD, and these animals the untreated animals get very severe rapid GvHD that leads to their death. Animals that are prophylaxed suboptimally with just rapamycin monotherapy do a little better but still die very quickly. And then the tacmethotrexate animals go on and have a much longer survival. Our GvHD looks just as we would hope it would histologically. We see T-cell infiltration in the liver, the GI tract, and the skin. And we do anonymous scoring from a pathologist who doesn't know the identity of any of the treatment groups, and you can see that we have really good correlation between our clinical disease and our pathological scores. One of the important things for the HIV community is we showed last year that the brain is also a target for graft versus host disease. Now, this can be a problem for our patients undergoing transplant, but says something about the ability of the T cells to invoke a graft versus reservoir effect even in the CNS. So what we showed is that in animals that got graft versus host disease, they had really a very clear signal of CD3 positive T cell, donor T cell infiltration. This shows the perivascular space and the parenchyma, and that this could be controlled but not totally taken away with either tacmethotrexate and rapamycin. I don't have the slides here, but I'll tell you that the T cells that are getting into the brain are CDH, not CD4. They are highly proliferative and they produce granzymes. We've gone on to look very carefully at other immune parameters that describe graft versus host disease, and this is data that we published previously using flow, which shows that when we look at the immune consequences of graft versus host disease, we see, first of all, a shift towards antigen-experienced phenotypes. We see cells that produce lots of granzyme and perforin. They become highly proliferative. They express Ki67 and pro-apoptotic. And we see evidence, and I'm going to show you this actually more in just a bit, of both Th1 and Th17 phenotypes. So we've done a lot of flow cytometry to look at the immune phenotype as we try to understand what these cells might be doing for graft versus host disease in the graft versus reservoir. But several years ago, we decided to take a more systems approach to this, and this really has led to some very interesting observations that I think are going to be relevant for HIV. So what we can do in our animals is do our transplants, and then at timed intervals after transplant, sort CD3 cells, we make RNA, and this has been a gene chip analysis because we started this sort of 5 years ago now, before people were doing RNA-seq. What we show is that this is a PCA analysis of our data, which just shows that we have really nice separation between our healthy controls. We can also show this with autologous transplant controls and our no treatment controls, and our different treatments really fall in different space, suggesting that our transcriptome is going to be correlated nicely with clinical data. And indeed it is. So those animals that aren't prophylaxed get this very severe primary, what we call primary GvHD, and it's really is skewed very much towards a Th1 response. So you see CD2 upregulation, IL-12 upregulation. Interestingly, for HIV, you actually see CXCR4 downregulation. This is the red here, and CCR5 upregulation. So those cells that are causing primary hyperacute GvHD are actually skewed towards CCR5 upregulation, which may potentially be a problem if you're thinking about potential for being newly infected. But that no therapy control is pretty different than what we see in patients, right? Because we never give patients no prophylaxis when they're transplanted. So without prophylaxis, these animals develop this primary GvHD. But what's likely more relevant to our patients with transplant and with HIV is that with prophylaxis, you don't get this primary GvHD, you get through what we call breakthrough GvHD. And this is likely the most relevant for the graft versus viral reservoir effect. And so what we did with those, we have a large group of transplants that had this graft versus host or breakthrough graft versus host disease, and then so we did a transcriptomic analysis where we compared them to 3 control groups. First of all, autologous transplants, the no therapy, so we wanted to take out those genes that are specific for primary GvHD, and healthy controls. And what that's led to is a group of 172 genes which define that breakthrough GvHD signature. And when we did pathway analysis with these, we found that they are very concentrated in, first of all, JAK-STAT signaling, and also the KEGG pathway known as cytokine receptor interaction. And when we probe this more, this is what we see, which is really interesting. So if you take a look, this is what we find is that those CD3 cells during breakthrough GvHD are enriched for Th17-related genes and not Th1-related genes. So what you can see here, the orange is the breakthrough, and you can see that you have upregulation of IL-17, IL-22, RORgamma-C, and AHR, all Th17-specific transcripts. And that's very different than the Th1 skewing that we see with the red, which is that primary GvHD. And again, really important for this community is that, importantly, CCR5 is actually downregulated on those Th17 cells. And this may be really a positive thing for our patients. So if the cells that are causing that breakthrough GvHD are also those that are causing graft versus reservoir effects, we may have a leg up because they probably are already downregulating CCR5. So they may be not only inherently more resistant to the virus, but they may also be more amenable to our gene editing approaches that just need to downregulate the expression of these entry proteins at least a little bit, potentially less than if you have a Th1 response. So we've developed a model now that really describes our thinking about this shift during graft versus host disease and, of course, during graft versus reservoir. So in autologous transplants, you get this proliferation of T cells during homeostatic proliferation that is not pathogenic. When we have this no therapy control, we get this very rapid Th1 skewing, which rapidly leads to death of the animal from graft versus host disease. But in the setting of prophylaxis, and this is what we see in patients, what we often see is this breakthrough disease. And this is not Th1 skewed, but rather IL-22, Th17 skewed. And we think that these are the target cells for the graft versus viral reservoir effect, and these should be the ones that we're really thinking about when we're thinking about eradicating virus. So in conclusion, we've developed an allogeneic transplant and graft versus host disease model in Rhesus macaques. We found that the GvHD targets are very close to what we see in patients, skin, gut, liver, and newly in brain. We find that it's associated with the cell and serum markers of T-cell activation and a complex gene expression program of activation, effector function, chemokine, cytokine secretion, and cell cycle progression. And importantly, during breakthrough GvHD, in the setting of prophylaxis, we see a progression from a Th1 to a Th17 dominance. And this leads us to a hypothesis that we'll be testing in our infected animals that Th17 and Tc17 cells, so these are the CD8 cells, may be key effectors for the graft versus viral reservoir effect and may represent the best targets for HIV-resistant gene editing. And finally, I was asked to really have a summary for the community, and since I'm a rule follower, I did this, and this I think was the hardest part of the talk putting together for me. So for members of the community that are here, so what were the key questions of research that we addressed? And really what my lab is really focused on is the question of how can we harness the immune system after transplant to help eradicate HIV? I think the key findings and take-home messages for you is that after transplant, T cells undergo a transition to favor those that we call Th17 type, and these may be the most important cells for the graft versus viral reservoir effect. The question of why is this important and how is it related to the cure? And this really becomes the hearkening back again to the Berlin patient, is that if we are to understand the reasons that Berlin patient was cured, we need to understand how his donor T cells may have helped clear the HIV reservoir. And so to do this, we have to understand how T cells are activated after transplant. And finally, should we or should we not be excited about this? Well, I'm excited about it. I think these experiments do get us closer to understanding the ways that T cells can target recipient cells after transplant. And of course, the more we know about this, the more we can engineer those T cells to clear the reservoir. And I think the other important point from our work is that they also show that donor T cells can get into the brain after transplant, and this may also be important in clearing the CNS reservoir. Finally, I'll just thank my lab. Jack Schell is here, who's leading our HIV eradication work, and the rest of the lab and the Primate Center, and of course Guido, who I did the first work with, and Hans Peter here in Seattle. Thank you.
Thank you so much. Are there any questions? Matt?
Yeah, I have a question for the transplanters in general. So, Dr. Ambinder made the point that we're probably doing multiple transplants a year with CCR5 negative homozygous donors without knowing. Now, do we know how those donor transplants do compared to heterozygous donors in terms of immune deficiency following transplant?
No.
You were saying, what were we doing with that?
We're probably using donors who are CCR5 delta 32 homozygous without even knowing them.
Not for HIV.
Not for
No.
HIV.
Yes, yes, you're absolutely right.
Or other diseases. Is there any data on how those donors do, how those transplants do using CCR5 delta donors?
There's a little bit of data, and I'm a little bit rusty, but thinking that it was conflicting, but overall there was no clear impact.
Yeah, I mean, I think, before the Berlin patient, people weren't tuned into this. But what we know is that patients from the Western European sort of registries, patients just walking around the street that are delta 32 are fine. What we don't know is whether this, along with the immune deficiency that we give patients after transplant, how this is going to interact. We don't know, and I'll tell you, because transplant itself is so complex, we're going to have to do a lot of transplants to figure this out or to retrospectively look at this. And I'm not sure there's a good way. It would probably be in Germany to really have them look back at their registry where they've, probably 1% of their transplants are used.
Studies is coming back now. So there was a suggestion there may be less GvH from some. There was not more deaths from CMV or
Yeah.
transplant lymphoma or anything like that.
I think David had a question.
I'm curious about the non-human primates. Whether their primates behave like the primates in humans, and attributes the same. And whether you control for that in terms of age in these transplants.
Right. So we do control for, in terms of age, so all of our animals are what probably would be called young adolescents. So they're young just because of the size of them. So we use animals anywhere from 3 to, let's say, 6 years old, which is sort of a young adolescent. What I know from doing lots of transplants actually with the reduced intensity conditioning, that so even with reduced intensity, we have a very hard time finding the thymus post-transplant, and we can find it pre-transplant. So we know that just like with patients, we are doing damage to the thymus from conditioning the animals.
So I think in the interest of time, unfortunately, we have to move on and move into the break. Thank you very much again.
Thanks.